Hello, everyone, and welcome to today's live broadcast, The Power of Multiplexing with Luminex Technology, presented by Dr. David Bourdon, PhD. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. To learn more about Thermo Fisher Scientific, please visit www.thermofisher.com. Thermo Fisher Scientific is the world leader in serving science with revenues of $17 billion and approximately 50,000 employees in 50 countries. Our mission is to enable our customers to make the world healthier, cleaner, and safer. We help our customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. Through our premier brands, Thermo Scientific, Applied Biosystems, Invitrogen, Fisher Scientific, and Unity Lab Services, we offer an unmatched combination of innovative technologies, purchasing convenience, and comprehensive support. I'm Brenda Kelly Kim, and I'll be your moderator for this event. Before we begin, I'd like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on screen. We'll try to answer as many of your questions as we have time for at the end of Dr. Bourdon's presentation. Also, please notice you'll be viewing the presentation in the slide window. To enlarge this window, just click on the screen icon located in the lower right. Finally, if you have any trouble hearing or seeing this presentation properly, please click on the support button at the top right of the presentation window or use the Q&A button lower left to let us know that you're having a problem. I'd now like to introduce our speaker, Dr. David Bourdon. David received his PhD in postdoctoral work focused on the study of G protein-coupled receptor pharmacology and thrombosis. Following his academic training, he led Luminex-based multiplex immunoassay platform development efforts at a Luminex partnering company. In 2009, David joined Thermo Fisher Scientific as a senior scientist working on the development of novel immunoassays platforms, Protoplex immune response assay. Working on next-gen immunoassay technologies, David is interested in working with translational investigators and key opinion leaders to identify serum-based biomarkers in cancer, autoimmunity, and inflammation. Beyond his research and development responsibilities, David is dedicated to platform strategy and the antibody content roadmap. I will now turn the presentation over to David.
Thank you very much, Brenda. Hey, everyone. It's great to meet you all. Thank you very much for joining our LabRoots event today. So as I was introduced, I lead an R&D team here in Carlsbad, California, focusing on next-generation immunoassays, as well as expanding our Luminex-based platform. Today, I'd like to give more of an introduction to the Luminex technology. Even though the technology has been out for a number of years, there's still quite a few people who are not as familiar with the technology and have yet to take full advantage of the power of multiplexing and how it can really save you time, money, and give you higher quality data. So let's get started. So first of all, I'll admit, from my doctoral work and thereafter, I really am a proteomics person. I really do appreciate the power that the whole genome sequencing efforts have uncovered, but really a lot of, I think you'll see in the next coming decades, a lot of the groundbreaking work will be in the proteomic space. Therapeutic monoclonal antibodies is a great example of how immuno-oncology has really taken off. And so this slide just kind of overviews or highlights that, post-Human Genome Project work, there's at least 20,000 proteins that have been identified, and of course that number keeps getting larger. The post-translational modifications and all sorts of subtle differences in protein modifications, acetylation, phosphorylation, etc., really can make a difference. And so this schematic on the right side is just to remind everyone that, when you're talking about, let's say, a kinase signaling pathway, there's a lot of different knobs that can be turned by our own immune system, our own cell biology, our own regulatory systems in our body. And this hits very close to home for me because, for example, my postdoctoral work is really focused on GPCR signaling. And I joke with my colleagues that if I had $1 for every time I ran a Western blot or wanted to look at protein modifications in signaling pathways, if I had $1 for every time I had to do that in a single-plexed approach, I'd be a rich man. And multiplex technology really helps interrogate a number of different protein pathways in one single experiment. And long story short, I wish I'd had the type of technology I'm going to talk about today. I wish I'd had it at my disposal when I was a postdoc. It definitely would have shaved some years off that work. So all joking aside, there's a lot of exciting work in the proteomic space. And so what I want to move on to here is really to talk about the key areas. I'm on slide 3, the key areas that our technologies focus on. And this is not all inclusive, but some key areas where we really are covering the space well are in the areas of inflammation, autoimmunity, and cancer. And as many know from the seminal work from Hanahan and Weinberg, that original paper came out in Cell in 2001, where Hanahan and Weinberg spoke of all of the different processes that are involved in the propagation of cancer. And this review article that I'm citing here at the bottom actually gives a very nice update in 2011 of what has been addressed in the last decade and where do we still have to go. And it's a very great read for those of you who haven't read it, but it really does outline how challenging cancer is to study, treat from the translational investigator perspective. And it also, it's very exciting for us in the biomedical research space, especially us in the tool provider space, because my job and the job of our team is basically to provide cutting-edge tools for investigators such as yourself. And so we really do, our sign of success is developing high-quality tools that are second to none that allow you to make great discoveries. So we're very excited about this work. We have, I think, as a community, there's a lot of work yet to be done, but we're on the right track. And so this wire diagram of the immune system basically, it's meant to show, and I apologize if it's too small to read, but the point here is that the immune system, our immune system is very complex. And we know that a single cytokine does not have a single, A plus B equals C responsibility. There are a number of cytokines that play roles in a number of different steps in the pathways. Everything from, let's say, activating a B cell to having maybe T cell function as well. And so being able to make sense of changes in inflammatory cytokines, for example, can be quite a challenge, especially if you were to do that on a target-by-target basis. And so the technologies that I'm going to talk about today allow one to take a single sample, let's say a human serum sample or a cell culture supernatant, and measure a number of targets in one shot. And I'll go over the reasons why you'd want to do that in just a moment, but you can imagine the type of work that is required to make these kinds of conclusions that are highlighted in this wire diagram, for example. And by the way, I forgot to mention earlier that we will be pushing out poll questions throughout this presentation, and please feel free to quickly select on your response. It will try not to make it too distracting, but I think it's great to get feedback from experienced users such as yourself as to your answers help us make better products. So thank you very much for that participation. So as far as creating clarity out of this chaos, I use chaos in a positive light. There's a lot going on in our immune systems. As we know, like I mentioned, therapeutic monoclonal antibodies are becoming a gold standard for a lot of treatment of diseases. In fact, last year I attended the American Society of Clinical Oncology meeting in Chicago, and it just amazed me the number of companies that one of their chief pipelines or their primary pipelines in the drug development process are all therapeutic monoclonal antibodies. And as anyone who has seen the commercials, you'll see a drug like Humira initially comes out for treatment of one disease, and now they're realizing that, you know what, a therapeutic monoclonal antibody against TNF-alpha can treat a lot of different inflammatory conditions. So it just shows the vast breadth of coverage that being able to regulate these inflammatory moderators can have on our health and our ability to combat disease. But being able to create clarity out of that chaos, and in other words, for us to develop tools that really provide the best toolbox for you, it all starts with antibodies. And what I've highlighted in this slide is basically just to remind everyone, the assays that we develop, they're only as good as the antibodies. And I speak very candidly about this because as a tool developer for a number of years, in fact, prior to our integration, and just for a little history, I started with the Invitrogen company, which eventually became Life Technologies, and then through a merger became Thermo Fisher Scientific. In the years prior to joining Thermo Fisher, it was quite frustrating to not have enough good, high-quality antibodies at my convenience or at my use, and so I wanted to build new assay platforms. But with limited antibodies, I really felt my hands were tied. And so, since the new Thermo Fisher Scientific organization, I highlight here that we have over 40,000 antibodies, and that number keeps growing. And I'm actually involved in our antibody roadmap initiative as well. And it's very exciting to see our work on developing new antibodies to exciting targets. I work with a very great team of antibody sensors who are working out with key opinion leaders to develop these best antibodies for new targets. So again, high quality kits and systems all start with high quality antibodies. Of course, we're going after the most relevant targets, for example, cytokines, chemokines, signaling proteins. But then also we want to be able to measure very low amounts of these proteins in all sorts of biological fluids, serum, plasma, and in fact, it's very interesting. We even have people coming to us that are studying, for example, dry eye disease or other challenging diseases where sample volume is very limited. And I'll get to that in just a minute. That's another reason why you would want to conduct as many measurements in a single well as possible, because some of the investigators may only have 20 or 30 microliters, or in some cases only 5 microliters. So they want to be able to get as much data out of that single reading as possible. So it's very important to be very customer-centric and make sure that we're listening to the end user and what they need most. And so one of my focus areas since joining the organization has been biomarker discovery and validation. And the term is used a lot, biomarker, but I think it's really important to understand that biomarker identification is not a single measurement. It's an entire process. And I've worked with a lot of great scientists currently and in the past that have taught me this biomarker space. And it's important to note here that, beyond studying the mechanism of a disease, biomarkers are really important to understand disease progression. So not only how a disease may start, but also to understand what to look for when a disease maybe is getting better or worse. And that's where translational investigators are very interested in the amount of circulating markers in, for example, human serum. And it's important to note, I'll only mention this once, but the products I'm going to be talking about today are research use only. But for example, I do have clinical investigators that I work with, which were taking these very disease-relevant samples and better understanding various levels of analytes in diseases such as bone marrow transplantation, for example, for blood cancers, as well as rheumatoid arthritis and other inflammatory diseases. The reason biomarker discovery is so important is because, as you can imagine, the samples from all sorts of individuals or even cell culture populations, they're very different. Not everything behaves the same way, and that's why biomarker discovery is so important, because a good biomarker needs to be able to be a protein or analyte that is known to be upregulated or overexpressed across a wide population. And what I'll show in just a minute is sort of a three-step process to discover, verify, and validate biomarkers. And it's very important to note that a good biomarker can withstand or survives the identification process and is robust enough to still be identified in orthogonal platforms. And I'll get to that in just a minute. But this is one area where I think that we really do set ourselves apart, that we take great pride in understanding this workflow and being able to know how to use the right tool at the right time. And that's something, I gave a talk at the American Association of Immunologists last year, and it was on general protein quant workflows. And the take-home message from that talk was, we have a lot of tools to use for protein quant at Thermo Fisher Scientific. And what sets us apart is knowing when to advise to a customer when to use the right tool. For example, when would you use flow cytometry versus Western blotting? Or why would you use a multiplexed immunoassay versus ELISA or Western blotting? It's very important to know why and be able to provide that guidance to the end user. So this is the biomarker discovery and validation workflow I was just referring to. And so it's a three-step process. And basically, if you look at this XY coordinate here, on the y-axis we have a number of targets. So imagine this is from 0 to, let's say, 20,000, the whole proteome or larger. And then this is the number of samples you would test for a given assay. So a discovery platform or a discovery tool is one that allows you to potentially identify hundreds or thousands of targets. Because of the platform, at least for now, and I'll get to this in the next slide, but you can imagine that tools like, let's say, mass spec, you're not going to perform mass spec on thousands of samples just because of the way the workflow is currently laid out. However, mass spec, on the other hand, can interrogate the entire proteome, looking for unique proteins that are maybe overabundant in a disease state. On the other hand, when you move from discovery to verification, what you want to do is you want to use a platform that you can test a larger number of samples, but maybe the total number of proteins it can measure is a little more limited. So you're basically casting a wide net in discovery and then going to something a little more limited in verification. And then finally, in validation, what you want is a platform that allows you to test thousands of samples, and this really gets to the point of statistical or clinical relevance. So the platform may be something that can only measure, let's say, 1, 2, or 3 analytes, but it's something that you can perform in high throughput or ultra high throughput. A great example of these 3 platforms would be, for example, Orbitrap. We are the world leader in this technology. And so this is a technology that allows you to truly interrogate the entire proteome. Once an investigator has basically narrowed down the potential possibilities from, let's say, thousands of proteins down to maybe 30 or 50, that's when you would use a bead-based platform, which is the heart of today's talk. You'd use a bead-based platform that you can measure a number of analytes. And then finally, once you distill this down even further, one might use a singleplex technology like ELISA. And by the way, we have a lot of ELISAs too. And so it's important to note here that whether you're doing discovery, verification, or validation, you got to use the right tool at the right time. Otherwise, you'll be wasting time, money, resources. And fortunately, we cover all three. Now, this is one area that I think in the future, a lot of people are looking to develop next-gen technologies that allow you to perform an immunoassay at the discovery level. And that would be another great talk in the future. So as far as focusing more on the last two, verification and validation, whether somebody performs a Western blot, a singleplex ELISA, or a multiplexed immunoassay really depends on a couple of different needs. For example, what's sensitivity? Do they really need the sensitivity, or are they just looking for a qualitative measurement? The antibody menu. Now that we have a much larger menu, that's less of a concern, but definitely, sometimes we only have a particular target in, let's say, a Western blot primary antibody and not a complete ELISA setup. So one might perform Western blotting for a quick answer. Throughput. Now, everyone knows that, for example, performing Western blotting on hundreds or thousands of samples can be quite laborious, whereas maybe performing a singleplex ELISA can get the same answer with much less headache. And then there might be other considerations too. A certain user may actually be wanting to confirm the size of the protein, cell localization, so maybe they're trying to separate organelle structures and look at whether or not a particular protein is co-localized with another protein, so IP might be important in that case, immunoprecipitation. So anyway, again, using the right tool at the right time is very important. And so let's dig deep or dive deep now into multiplexed. But I'll start out by setting up this example here. We all know sandwich ELISAs. The way they work, you have a capture antibody, typically a monoclonal antibody immobilized on a plate surface. You capture the analyte of interest, maybe from human serum, and then you'll come in with after multiple washes, you'll incubate with another antibody against the same protein, and in some cases, you might even use a secondary antibody. You can see there's a number of steps here, and of course, for the analytes you're trying to measure, you have to have each capture antibody on a different plate, because there really is no way to distinguish the reporter signal that originated from capturing one analyte versus the other. So, of course, people separate their ELISAs based upon the capture molecule, capture affinity reagent. Now, again, it becomes a little more challenging if somebody wants to look at a number of analytes. Imagine this situation where somebody wants to look at, let's say, not 4, but 16 different analytes. Well, that's a lot of plates, and you can imagine, wouldn't it be nice if you could compress all that analytical power into a single plate? And that's where we are going with Luminex technology. And so again, here's the traditional ELISA, and then here is the backbone of Luminex. So Luminex Corporation is a company that started in Austin, Texas, and their core intellectual property is around the ability to manufacture these 6-micron polystyrene beads. What makes these beads special is that the beads are dyed or colored with a ratio of infrared and red dye. So what we're seeing here, just for example, is an 8x8. So 8 different concentrations of a red dye and 8 concentrations of an infrared dye. And you can imagine that this is, for example, low, low, and this is high, high. What this ratio metric combination of these two dyes allows one to do is to be able to identify which bead is which. These are fluorescent dyes, so you can imagine excitation, the emission spectrum given off by this bead here is a very different readout than this bead here. And so what that allows you to do is to basically utilize different solid phases for a ELISA-like technology, but now on a bead. And so here's, again, the same 8x8 example, but let's now pretend that In this case, and this is the way our kits behave, one would conjugate an anti-IL-2 antibody on this bead region, and they're called bead regions to basically identify each one as a unique bead color. So in isolation, we conjugate this bead region to an anti-IL-2 antibody, this one to an anti-IL-4, GM-CSF, and TNF-alpha, for example. And these beads, these 6-micron polystyrene beads, have about 3 million binding sites. So it's not just a single antibody. It's many, many millions of antibodies on the surface of the bead. And what happens here is now you've got, in this case, we have 4 different bead regions, each conjugated with their own analyte-specific antibody. But now what we can do is we can combine these beads in a single well. And because each antibody, each bead is conjugated with a very high quality antibody that's very specific for a particular analyte, the IL-2 bead only binds IL-2, IL-4 bead only binds IL-4, etc. So after a wash event, so first the beads are combined, then you add your analyte, for example, human serum. You capture the analyte-specific events with the 4 beads, and then you wash the beads and then come in with another set of antibodies, a cocktail of detection antibodies. And all these detection antibodies, again, are specific for the particular analyte of interest. But what's unique about these antibodies is they're already pre-conjugated with a fluorophore phycouricin. And this fluorophore is in common with all of them. So basically, this is the reporter signal. So imagine here, each one of these beads is identified by its fluorescent readout for association with a particular analyte, and then the reporter readout is proportional to the amount of binding that occurs on the surface of the bead. So what you can see here is that larger reporter signal equals larger number or larger amount of analyte in the sample. So again, the RPE quantitates the binding events, and the bead region identifies which particular analyte you're measuring. And there are 2 types of instrumentations two different types of core technologies that Luminex uses to identify the beads and measure the reporter. One is a flow cell-based system, which some of you in the lab might have seen an LX100 or 200 instrument or a FlexMap 3D. The other, more recently released instrument is basically a planar flow cell. And what happens is this is limited to the magnetic beads. When the beads enter the flow cell, a magnet pulls them down to create a monolayer. So now you're actually exciting and measuring the emission of the beads for the red signal, which of course gives you, for example, like a microarray layout of all the beads in monolayer. And then the green laser comes in and identifies the amount of binding that occurs. So a similar concept, just a different way of looking at it. So in the end, what this technology allows you to do is, just for the sake of our initial example of looking at these 4 analytes, this is the way you would have to measure these single analytes using ELISA. But now this is what you can do with Luminex-based technology. In a single well, you can measure all 4. And this is just for simplification of the example, but you can imagine here, we have about anywhere from 3,000 to 4,000 beads per well for each analyte. And so what you can imagine here is there are many, for example, thousands of bead number 1, thousands of bead for bead number 2, et cetera, et cetera. So not only are you able to measure each of the 4 analytes in this example in one well, you're measuring a lot of different binding events, whereas in an ELISA, you're looking at a single readout, a single snapshot of the bottom of the planar microplate surface. And so the way the instrumentation works for Luminex is each bead, and this was kind of highlighted here, each bead comes down the flow cell sequentially and gets interrogated by both forms of light, either laser or LED. But there are many, many binding events, or many events measured in a Luminex-based platform versus an ELISA. And on the right-hand side here, I've just very quickly laid out the workflow in terms of starting with the antibody-coated beads all the way through acquiring the data. And by the way, this slide deck, as well as our white papers and other brochures that highlight this in greater detail, are available through our site. So a great example of why, or the power of this is, for example, with AKT. We have an AKT 7-plex that allows one to measure 7 different targets involved in AKT signaling simultaneously. So you can imagine here that, and this kind of brings me back memories of my postdoc where, I might have been looking at a phosphorylation pathway, but looking at it one protein at a time. And if that one Western blot didn't work, or I identified, wait, okay, it's not that protein that's involved in this particular pathway, I would then have to go back and try another protein candidate. Whereas with multiplexing, you can cast a wider net and cover a lot of bases in a single assay. And so this next image here really does highlight one of our newest launches, and that is the Cytokine human 35-plex. And this is just an example of the wealth of data. So this is across 8 different samples, 1 through 8, and then looking at all of the different cytokine profiles for those 8 samples. So it's kind of a nice snapshot of where some targets, and honestly this graphic here, it's kind of hard to see very subtle changes of these other lower bars, but you can imagine that certain targets are significantly different in certain samples, and others are basically at basal or constitutive levels and don't change at all. So again, one of the highlights of the power of multiplexing is, reduced cost per data point, reduced labor time, reduced sample usage. Again, 35 different analytes measured in a single well, and this is from a 25 microliter serum sample. Increased analyte to analyte, reduced analyte to analyte variability. Again, if one was to measure all of these 35 different analytes in isolation, somebody would have to go back and put all that data together and make sure that all the controls matched up. And I've heard of people trying to normalize data from dozens of different ELISAs, and you can imagine if in one particular readout they add a little more sample than the other, it just creates a lot of headache, and all of that headache is eliminated in multiplexing. And then finally, the amount of data you get per sample. Again, I mentioned some people that are looking at human tear samples or very small, maybe in the case of a mouse cytokine study. We all know the terminal bleed for a mouse is a very small volume. And so one can imagine that they want to get as much high-quality data out of a single sample as possible. And then finally, this inset here just provides a nice summary of the cost per data point. And it's kind of hard to read here, but the cost per data point for ELISAs is in the blue here. And then I just outlined in red the cost for the analytes in Luminex. So it's a much more cost-effective approach than single-plex ELISAs every time. But again, it's about using the right tool at the right time. If one has gotten to the point where they have narrowed their biomarkers down to just 1 or 2 targets, ELISAs or another single-plex technology is perfectly practical. I mean, it's well suited. However, somebody is still in that targeted, I call it targeted discovery. If they don't have a higher plex, like Orbitrap technology at their disposal, even a 35-plex could be considered a more limited discovery or a verification study. So if they're at that point in their workflow, they definitely need a Luminex assay workflow platform. So this is just a great example. And by the way, again, I mentioned this is from one of our white papers. We have these white papers available for you to download after this talk. But this is just a great example of the great precision. And this is from our human Cytokine 14-plex. And so this is plate 1 in red and plate 2 in gray for a number of different analytes here. And you can see the plate-to-plate variation is very, very low. In fact, it's under 10%. So this is, again, another reason why it's important to focus on bead-based Luminex assays for this discovery work, because that plate-to-plate, inter and intra-assay variation, we want to minimize that as much as possible. And our Luminex assays perform very well. This is another slide from that white paper just demonstrating the dynamic range as well as the concordance. So on the left-hand side here, this is the dynamic range for a particular target, looking at it with the Luminex assay here in blue. I just highlighted it in red. And then this green line over here is basically a more limited dynamic range of a corresponding ELISA. And the importance of dynamic range is, of course, fewer sample dilutions are required and fewer repeats. I think for anybody that's worked in the immunoassay space, sometimes you've run some samples and they're either, typically they're too concentrated. If they're too dilute, some people will try to concentrate their sample, but that's where, if the sample's too dilute, that's where having a very high sensitivity assay can come into play. So we'll in the future be talking about aspects of those technologies as well. But in the case of this dynamic range, it's really important to not have to repeat assays because, let's say, the sample is too concentrated. And in the case of an ELISA, sometimes the sample will be out here somewhere, and it's too concentrated to be read on the platform. But that's where Luminex technology, because it has a broader dynamic range, can be very helpful. Secondly, concordance. A lot of people will be switching from our ELISA platforms to a Luminex platform. It's really important for them to know that a measurement that they took on the ELISA platform compares or concords well with our Luminex platform. And this is just a great example of an analyte where we're looking at it on the y-axis using a ELISA platform and on the x-axis with an Invitrogen multiplex. And the concordance is a nice R-squared, very high R-squared value. So it nicely concords. And what that does for people is that it allows a smooth transition to Luminex technology. It also is nice to compare data sets. I think I've talked to a lot of people that have used different technologies before, and sometimes there's a multiplication factor, and they basically have to sort of transform the data from one platform to the other in order to compare it. And that always makes me a little apprehensive because it means that there's something fundamentally different in the assay that is possibly measuring something different in each assay or each platform. In our case, we see great concordance between ELISA and Luminex assays, and that's really important. Also, it's great for cross-site collaborations. We want people to feel comfortable to share data with different individuals depending upon what tools you're using. So this next slide was really just to give you a couple quick examples of how our customers have used our technology to benefit their work. A lot of you may know of this study. This was the classic TGN 1412 trial study. Over a decade ago, there was a small biopharma company in the UK that was developing a therapeutic monoclonal antibody against CD28. It was considered a T-cell specific antibody, and it lacked a co-stimulatory requirement. And the antibody was developed to target autoimmunity and chronic lymphocytic leukemia. It was designed to bind CD28 and release anti-inflammatory cytokines and expand Tregs. When this therapeutic monoclonal antibody went to clinical trials, there were adverse events, but people were harmed from it, and it was quickly taken off the market. Well, this group here, this group actually, our customer, this was not the group that developed the antibody. This was the group that actually did a retrospective study, and this was published in 2010. But they basically went back and tried to better understand what happened and what possibly could have led to these adverse events. So what they did is they used our 30-plex Invitrogen human cytokine panel in a monocyte culture system where they added the TGN 1412 or the CD28, anti-CD28, and then measured a number of different pro-regulatory cytokines and chemokines. And what they found was that upon addition of the TGN 1412 or the CD28 antibody, it basically caused recruitment of monocytes to the vascular endothelium, which then caused a release of a number of inflammatory cytokines and, most importantly, recruitment of natural killer cells. And so over the years, we've seen the term cytokine storm used in a variety of different contexts. And this is a great example of how it's believed now that this therapeutic monoclonal antibody was actually doing a lot more than originally intended. And so this is just a great example. I'm not saying that this could have prevented what happened, but it was a great example of how we can learn from particular situations. And I think now a lot of people are doing this type of multi-target examination in a proactive manner to really understand the system before it moves on in phase trials. And you can imagine this would have been very difficult work to perform on a single-plex target-by-target basis. Okay, this next study, this is near and dear to my heart just because of my interest in systems biology and high-content screening. This study was an interdisciplinary group that used our 25-plex cytokine panel to understand chronic lymphocytic leukemia. So CLL, if many know, is a combined disease, a disease that is believed to start as an inflammatory response. And so what this group of authors did is they looked at 84 CLL patients and 49 age-matched controls. What they did is they also knew of the patient populations and how severe their form of CLL is. And they scored the severity of the disease as either their outcome was sort of abbreviated as good or poor. So an individual that needed therapy in less than 5 years after being diagnosed with CLL were giving a poor outcome. And that dendrogram here is basically indicated by the black boxes. So these are individuals that had a more severe form of the disease. The individuals that had a less severe form of the disease or a good prognosis were individuals that did not have to receive treatment until greater than 5 years after initial diagnosis. And those individuals are highlighted in the blue here. And then healthy age-matched controls are indicated in yellow. And again, let me just remind everyone, again, that our assays are only for research use only. This is not used for any clinical diagnosis, but it's very insightful and very informative to understand how these disease-relevant samples have varying levels of these cytokines. And so what the authors did is they took all of these human serum samples and they looked at the 25 different cytokines. And what they found was varying levels of about 7 or 8 different cytokines. And what we see here is that they've clustered the cytokines based upon these levels. And this heat map here, basically from low to high, you can see here from blue to red. This is basically the 0 point is right down the middle. A blue is a decrease in expression. A red is an increase in amount in the sample. And what you see here is the authors identified a cluster, they call cluster 1, that appears to be elevated in the individuals that have the most severe form of the CLL. Whereas there are 2 other clusters that stand out that appear to be more associated with individuals that do not have the most severe form of the disease. And again, this is not the be-all, end-all study, but I believe that this is a really nice discovery study to outline where we may start looking for commonality in this very awful disease. And it may be important to understand what's the root role of these cluster 1 cytokines versus clusters 2 and 3. And as we already know, therapeutic monoclonal antibodies are being used in a number of different treatments of inflammatory diseases and cancer now. So one could imagine that a study like this could actually be a very nice foundation for new approaches for a disease like chronic lymphocytic leukemia. So let me just go through one last little bit of this presentation, and it's really to expand what we can do with the Luminex portfolio. And so what I've been talking about thus far is really how we can measure a circulating antigen. And this was basically my former manager. He and I, when we were working, we kind of coined, okay, in the serum proteome, there are two major components. One is, of course, all the inflammatory agents and chemokines and cell-to-cell mediators. And the other, of course, is immunoglobulins. And autoantibodies are an area of increasing importance. And autoantibodies, in short, and by the way, there are a number of great papers. Some of the leading experts in this include Karen Anderson, Josh LeBaire, and these individuals are really leaders in the area of autoantibody work. But autoantibodies are basically IgG molecules that are basically produced by our own immune system in response to the increased presence of a particular protein. It can be a tumor protein, another protein that may be upregulated in the development of a disease, but the point is that it's a human IgG molecule that is actually responsive or binds to the autoantigen. So what we've done with this protoplex platform, and that's what this is on the right-hand side here, we've developed a Luminex-based platform that instead of capturing an antigen on the surface of the bead using a capture antibody, we start by immobilizing the autoantigen on the surface of the bead. So now our affinity reagent on the bead is really the autoantigen or the protein. And then what we're doing is we're pulling out of the, for example, human serum sample, an IgG molecule that is highly specific for the autoantigen. And then what we're able to do is detect that human autoantibody with an anti-human IgG that, oh, by the way, has the same RPE fluorophore as we would use in our classic cytokine profile assays. So in one sense, it's a very similar layout. We're using the same readout system. We're just starting out with a core, the core bead has a different affinity reagent on it. And so what we've done is, in the past, we've actually tried to demonstrate, I think we've done a nice job of demonstrating the value of not only looking at cytokines, but also autoantibodies. And by the way, we have a very powerful microarray platform called ProtoArray, which it's about 23,000 spots, almost 10,000 human proteins spotted on the surface of the bead. And that's a great example of a high content discovery tool for autoantibody discovery. But in this case, what we wanted to do is we wanted to demonstrate, is there power in measuring not only cytokines, but autoantibodies in disease-relevant samples such as lupus. Many of you in the rheumatology field know that lupus is a very challenging disease to study because there are a number of different readouts. People look at different profiles, but there's no one gold standard readout to diagnose or to identify lupus. And so what we tried to do here was to better understand the value of looking at not only cytokines, but autoantibodies in the same sample. We ran these two different kits. So of course we have our 30-plex cytokine panel, and we have this other kit we call the protoplex autoimmune panel. And so basically it's a Luminex panel that has 19 different known autoantigens on it. And what you might notice, so people in the autoimmune field will recognize that not all of these proteins are limited to one particular disease. Some of them have to do with thyroid autoimmunity, some of them have to do with lupus or connective tissue disorder, etc. So this is more of a discovery tool. It was more of a proof of concept to see if there's value in measuring these two together. And what we see here is that this is a great example of looking at a number of SLE samples as well as healthy age-matched controls. And this is a study that was performed by a number of people in our group. And so it's a nice comprehensive study to show that we were able to identify a number of cytokines. In fact, I'm only showing 3 for this example, but we were able to identify 5 cytokines and 9 autoantibodies that are significantly elevated in the samples from those suffering with lupus. But the question here is any one of these samples the gold standard to be measuring? Well, the answer is no. If it was that easy, I think lupus would be one of those diseases that would already be conquered, and it's a very challenging disease. So what we did is we tried to identify, this matching matrix here basically identifies true positive, true negative, false positive, and then false negative. What we did is we started with 92 lupus samples and 85 age-matched controls, and they were well-curated samples, so we knew that this came from individuals that are suffering from lupus. And so we know that these are SLE positive samples, and the age-matched controls, we know that they are healthy and not suffering from SLE. And so what we have here is this matching matrix showing about 98% identification of, or 98% predictive of the lupus situation. However, that's 5 targets. And for anyone in biomarker discovery, we want to get that number as small as possible. The output or the outcome was a little less impressive with just the autoantibodies by themselves. It was only 80% predictive of the disease. But actually, and this is some beautiful work from one of our biostatisticians, John. And so it's important to note here that if you look at the 2 data sets together, not just 30 profiles or 19 profiles, but if you look at the 2 together, there's about 98% predictive power with only looking at 1 and 1. The work that we're still trying to validate is whether or not you can then further identify lupus from other autoimmune disorders using the same kind of panel. And that's work yet to be released. So it's just a great example of the power of looking at not just one type of signaling or one kind of protein molecule, but expanding the scope, especially in the case of inflammation and autoimmunity. One can imagine, again, if we go back to that wire diagram that I showed at the beginning of the talk, the immune system is a very complex system. And one can imagine that if you're not only measuring the cytokine family, but also autoantibody, for example, that's coming out of activated B cells, plasma cells, one may be casting a wider net on the full extent of the immune response. So this next slide is just to kind of remind everybody that we have a wonderful global field application specialist team that can help you with the assays that we provide. And so not only can they provide training and seminars, just like the one that's being presented today, but they can also help you with applications, troubleshooting, and this email address here below will take you directly to our technical support team, and they basically funnel out or disperse out the requests to a particular individual. We have individuals covering the globe. And so again, I mentioned earlier in the talk that we do have a nice white paper that really goes into greater detail than what I was able to provide today. But there's a lot of great things happening in the Luminex space. And again, we're one of multiple Luminex partners. You might see other Luminex kits from other kit providers, but we really do pride ourselves in the process, the quality, and our post-sale support that we provide to really go all the way from sample to answer to give you the best experience. And this slide here is just to kind of remind everybody of the diverse area. I was only able to talk about Luminex multiplexing today, but when you think about immune profiling or protein quant in general, we cover the gamut. Everything from all the recombinant proteins and antibodies that we offer. Didn't even get to mention superclonal antibodies, which are an amazing product for us. Not to mention all the ELISAs we sell. And again, as a tool builder, it's very nice to see that we're really expanding our menu. Again, with the Luminex portfolio, as I talked about today, we can not only measure cytokines and intracellular molecules, but also autoantibodies. I believe Jolene later today will be talking about our Attune focusing cytometer. And then I believe earlier today, we already presented our Western workflow that can now be done in less than 4 hours using our everything from separate, transfer, detect with iBond. And then finally, I just briefly touched on the proto-array human protein microarray. But the point here is that we have a lot of great tools. And again, the point is that use the right tool at the right time, depending upon whether you're doing targeted discovery all the way up to biomarker validation. And so with that, I'll open the line for any questions. Thank you very much for your interest. Hopefully this is just the first of other communications we have with those in the lab root space. We really look forward to working with you. Like I said, as a tool provider, one of our greatest joys is being able to provide tools, assays, and kits that really delight you and allow you to make those important discoveries and publish those discoveries in top-tier journals. So all of my coworkers, we really live vicariously through the success of our customers who use our tools. So we really appreciate your support. We really look forward to working with you more. And I'll open the lines for any questions you may have. Thank you very much. Thank you very much.
Thank you, David, for bringing us that informative presentation. It is time for our Q&A. I just want to remind our audience, if you'd like to ask a question, just click on the green Q&A button on the lower left of your presentation window. Type your question into the box that appears on screen and hit send. And we do have our first question already. How do you know that the protein targets in a multiplex assay are not going to have cross-reactivity and give you false data?
Great question. Okay, great. Great question. And in fact, I knew that was probably going to be something people would ask, and I actually have a slide for that because I have been asked that before. And when I didn't have this slide, it was very awkward to explain it because it's a very simple study, a very simple experiment. But trying to explain it without this slide is a little more challenging. But basically, what our R&D and manufacturing teams do is, this is a great example of a cytokine 9-plex. And what we've done here is we have each of the bead regions that are conjugated with an anti-IL-10 antibody. Let me put on the drawing tool. IL-10, IL-13, etc. And what we're showing here in green is the specific fluorescence intensity that comes from measuring that IL-10 signal with an anti-IL-10 detection antibody. And so one can imagine that if this IL-10 capture bead picked up other things, and by the way, so the detection antibodies are a cocktail, right? It's, in this case, it's a 9-plex, so all 9 detection antibodies are all in one vial. And so if the IL-10 capture bead also picked up, let's say, IL-13 or IL-4, you'd see those signals light up too, because of course the detection antibody for IL-4 and IL-13, they're all in the mixture. And the point here is you see a very nice signal-to-noise ratio. So again, for those that can't read, because I'm sorry, the slide's too small, but this IL-10 specific signal is 6,780 fluorescent units. And the background signal is single digits, 8, 9, 8, 8, 9, 8, 9, 9, median fluorescence intensity. And so you're talking 6,700 versus 9. So that's a huge fold signal to noise. And what you see that is you see that same pattern all the way down the 9 targets. Well, this type of measurement, this type of test is run on all of our cytokine panels. So you can imagine for our 35-plex, we also have this same type of graph, but it covers all 35. It's just, I didn't put it up here because it would go off the page. it would be too large. So, but it's a great example of we just demonstrate that there is specific signal. And again, it all depends upon the high quality antibodies and making sure that those antibodies don't cross-react with either antigen or other antibodies. And that's a key point to mention here, is that one can, so you have to worry about not only antibody-antigen interaction, but antibody-antibody interaction. So I hope that helps answer that question. Are there any more?
Yes, we do have another question, and we're coming up on time. This is going to have to be our last question, but an attendee asks, are the Luminex assays more sensitive than ELISA?
Thank you. Great question. So it really depends upon the target. Some of our targets, and by the way, this is all readily available in our in the product insert. Some of the targets actually are more sensitive. It really depends upon the target. We've seen some that are actually, it's a great antibody pair, but it really depends. It's a target to target. You can imagine, though, the number of antibodies to choose from when you're looking at, let's say, a 35-plex, the most important specification is to make sure that there's no cross-reactivity whatsoever. And so if we have, let's say, we test 10 different antibodies, some of them may actually be more sensitive than ELISA, and they could produce a more sensitive assay. However, those antibodies that are more sensitive may have cross-reactivity characteristics, and we then choose not to use that antibody. Even though it might be more sensitive, we would choose not to use that antibody because we do not want cross-reactivity in a plex panel. So it really depends on the analyte. But there are some that are more sensitive, but I do want to say with confidence that they're on par. Yeah, again, it depends upon the analyte.
Okay, thank you for those answers, David. We are out of time for our questions. I just want to ask Dr. Bourdon if he has any final comments or thoughts for our audience today.
No, well, I just want to first of all thank everyone. I guess later I'll find out how many attended the talk, but this is a new platform for us. It's very exciting as a scientist leading a group. We definitely, I love the interactive nature of this. I hope we can do it more in the future. I hope that some of you hopefully will be talking to you in the future. It allows us to really get our message across, and we just look forward to working with all sorts of individuals looking for the best tools in protein quant. So hopefully I'll be speaking to some of you again in the future, but please feel free to reach out to our sales force, our field application specialists. All of them know how to get a hold of me. So if you have any particular questions, but any of us can help you, and we really look forward to empowering you to get the best results.
Thank you too, as well, David. It's been great having you here and having you bring this important information. I want to thank also our sponsor, Thermo Fisher Scientific, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June of 2017. You'll be receiving an email from LabRoots alerting you as to when the webcast is available for replay, and we would invite you to share that email with any colleagues who may have missed today's live event. That's all we have for now. Thank you for joining us, and we hope to see you again. Goodbye.